Hello, I am Dr. Hannah Cox from Prevention Genetics and Exact Sciences Company. Um, I work as a laboratory director doing genetic testing primarily for hereditary cancer. I want to thank you for inviting me to Male Breast Cancer Happens. I attended this virtual conference last year and I absolutely loved it. So, thank you for the opportunity to attend again. And today I'd like to talk to you just briefly about germline genetic testing for inherited cancer with a focus on male breast cancer. Just to begin, I do have to go over some brief disclosures. This presentation is sponsored by Exact Sciences, for which I'm an employee and also a shareholder. And the content's really going to focus on one of the products at Exact Sciences, which is Riskard. And just be aware that the information presented today isn't clinical, diagnostic, or treatment advice for any particular patient. With that, let's get started. Let's talk about the statistics of male breast cancer. I touched on this briefly last year, and the statistics really haven't changed aside from one major one, and that is the estimated number of new cases in the US. Last year, it was estimated at about 2,500. The American Cancer Society actually estimates 2,800 men will be diagnosed with breast cancer in the US in 2023. Now, when we talk about mortality and morbidity, approximately 530 deaths in the US are going to be attributed to male breast cancer in 2023. This represents 1% of all cancers in men and 1% of all breast cancer cases in the United States. And one of the key differences between male and female breast cancer is that the average age of diagnosis is much higher in men. It's in the late 60s. And we know that the incidence is approximately 1.1 males versus 126.5 females per 100,000. Now, there are a lot of factors that contribute to an individual's lifetime risk of male breast cancer. These factors can include, if we start on the right hand side of this circle, um, increasing age. As we talked about, the average age of diagnosis is late 60s. How healthy you are. Whether you have an increased weight, how much you exercise, the type of exercise you do. Diet and lifestyle factors are also a big part of this. How much alcohol you consume and the type of diet that you consume. Exposure to radiation can also increase your risk, and exposure and levels of certain female hormones, such as estrogen and progesterone. Now, some additional major factors are actually your family history and also your individual genetic profile. Both of these can actually significantly increase your lifetime risk of predisposition to male breast cancer. Focusing on that family history and genetics, these are two key risk factors, and there is a bit that we know about these. In terms of the genetics, approximately 4 to 40% of male breast cancer will be due to inherited genetic changes. The reason that risk is, or that percentage is so wide, is because there's still a lot we don't know about male breast cancer, and it depends on the types of studies that are undertaken, what genes they're looking at, and in what human populations they're targeting. And so at the moment, we still don't know a lot about it. But what we do know is genes like BRCA1, BRCA2, CHEK2, PALB2, and several others can actually significantly increase your lifetime risk of developing male breast cancer. There's also an increased risk too of individuals that have Klinefelter syndrome, and this is typically a man that has an additional X chromosome. So, aside from this genetic background, family history also plays a significant part in determining what your lifetime risk may be. What we do know is that if you have a family history of hereditary cancer, particularly breast cancer, you have an approximate 2.5-fold risk of developing male breast cancer. And this is supported by 20% of men with male breast cancer having a first degree relative with breast cancer. And as the number of affected breast cancer relatives, especially with early onset breast cancer increases, we know that your lifetime risk of male breast cancer increases as well. And this can be greater than a fivefold lifetime risk compared to general populations. So, one of the options to actually really dig into those two risk factors, your family history and also your genetic background, is through genetic testing. One testing option is this Riskard hereditary cancer test. Now, this is a multi gene next generation sequencing panel. It screens for not just male breast cancer, but a whole spectrum of adult onset cancers. And why do we want to look at adult onset cancers? It's because some genes like BRCA1, they're actually associated with an increased risk of multiple types of cancers, including male breast cancer. And so by having more genes on the panel for a targeted set of cancers, you can actually be more comprehensive in your genetic testing. Now, this test and with the content is designed to be clinically actionable. It's evidence based, which means the genes on the panel have strong associations or strong knowledge behind them of hereditary cancer and medical management and surveillance strategies that are recommended by professional societies or through scientific research. And the idea of having a panel is you minimize uncertainty. You're trying to ensure you have the genes with the most meaningful information for the patient at that time point which they're getting tested. Now, this test also provides an individualized report which looks at a patient's risk of not just male breast cancer, but potentially other types of cancers depending on their molecular test result. And for positive test results, it will provide information about risk to family members and also care options, including surgical or surveillance or screening strategies that can be discussed with the patient's healthcare provider to personalize their journey. Now, last year I talked to you more about the report and the outcomes of genetic testing and a patient's story. And this year I want to talk to you about how we actually run these tests in the laboratory. And so, if we break it down into main steps, there's five. We start off by extracting DNA from the patient's specimen that we receive in the lab. We then have to go through and actually sequence those genes that we're targeting, and we call this sequencing. And we do that on a big machine called NovaSeq 6000, which is designed by a company called Illumina. We can get all of this really valuable genetic data for that one particular patient that we've extracted the DNA from, and this is represented as reads of what we call nucleotides. And nucleotides make up the individual units that make up DNA, and we represent these as A, T, G, and C. And so, as a geneticist, what I'm looking for is any changes in the patient's DNA compared to what we see in general human populations, which we call a reference genome. And then my job is to determine whether those changes are just benign ge- general changes that are present in the population, which are typically interpreted as benign or likely benign, or whether these individual changes are rare, and whether there's additional information there that can tell me whether these are actually clinically actionable, which is what we interpret as pathogenic or likely pathogenic. Occasionally, what we find is rare variants where there just isn't enough information for us to say whether it is definitively clinically actionable or not clinically actionable, and we refer to this as a variant of uncertain significance. That information is then collated together into an individualized report, which will have a broad summary statement or summary of findings, which may be positive or clinically actionable, indeterminate, negative, and occasionally we might have other findings because genetics doesn't like to play by the rules and everybody's unique. So, starting at that first step, genomic DNA, just about every cell in your body aside from red blood cells, has what we call a nucleus. And inside that nucleus, which is the cell powerhouse, is where your genomic DNA is stored. So, to access that genomic DNA, we need to extract or get those cells into a solution, and we want to lyse them, which means getting the cell membrane and the nucleus to release all of that genetic material. Now that genetic material is suspended in liquid, we want to separate out the genetic material, the DNA, from other components in the solution, such as proteins. We then precipitate this out, so remove all of the solution. So all we have is that beautiful genomic DNA. We then want to purify it to make sure we have the highest quality for that patient. And then we review that quality in terms of the volume we've extracted, the DNA concentration, how pure it is, and whether it's maintained its integrity. After we've extracted that DNA, the next step is to actually fragment that DNA, because you have in every cell of your body two strands of DNA, each that has 3 billion nucleotides in it. It's very, very big. And we only want to look at a very, very small part of that, which is those individual genes. In order to access that information in those genes, we fragment the DNA into smaller pieces, and then we add these little sticky bits to the end of those fragments called adapters. Those adapters are then used in solution and are amplified over and over again, because remember, DNA is undetectable to the human eye. So we need to get it to a level that's high enough that our machines, even though our human eye cannot see it, our machines will actually be able to detect the presence of that DNA in the specimen. We then use this really nifty technology called a flow cell. And essentially, it's a glass slide that has little sticky bits of oligonucleotides, which is just a type of DNA. And we hybridize or attach the DNA fragments from that patient with the adapter on the end of it to a complementary sequence that's attached to a glass cell. And this glass cell is just absolutely tiny. And all of those fragments are lined up on there and stuck to it. We can't even see it. We then do a reaction where we amplify and make tons of copies of all of those DNA fragments in that glass flow cell chamber. After we do that, we then have a laser that runs through and starts at the bottom of that glass flow cell and then goes up the individual nucleotides within that strand of DNA and reads every one of them and generates a sequence for me to read as a geneticist. These short sequences come across as fragments because remember, we did fragment the DNA. And so the next thing we need to do is realign all of those fragments using informatics and analysis pipelines to what we have as a generic reference human DNA specimen. You're seeing this up the top here. And if we align all these fragments over, because those nucleotides create a really distinct pattern, and we can see here in the orange, this particular fragment in this individual, they actually have a C instead of a G nucleotide, which is what we see in general populations. And so this is where I need to come in as a geneticist and look at it and determine whether this change is actually clinically actionable or not. So, in terms of interpreting sequence variants, there's a lot of standards and guidelines out in the field for how this is done within a professional clinical setting. We have a lot of information that we have to pull together to look at that change from the reference human DNA sequence. We want to look at the type of the variant and whether it impacts the encoded protein, which is the biological product that is produced by your DNA, or your DNA tells your body to produce within the cell. We want to look whether there's information of whether this variant actually is transmitted So we look at the frequency of the variant in individuals with disease, the frequency of the variant in individuals without disease or healthy individuals. We use computational models that get in there and look at the molecular structure of DNA at that particular position and tell us whether we can predict that it's going to be damaging or not. Um, we look at whether there's other clinically actionable or pathogenic variants at that same position within the genome. And then, if we're lucky, there'll be scientific evidence and experimental studies, such as in mice, in cell lines, in zebrafish, in other animal models, enzyme assays that will actually give us information about whether this genetic change actually impacts the machinery of the cell. We then interpret that variant in one of these five main categories, which I talked about before, which is benign through to pathogenic. Pathogenic being clinically actionable, benign being not clinically actionable. So, to look at an example report, if we have a positive finding in this gene, BRCA2, I go through and I look at all of that information. And in this case, I can see this variant results in what we call a frame shift, which means it causes a premature stop signal for the cell making the protein. And so instead of expecting a protein this long, we may get a protein this long. Shorter proteins in this gene typically are not functional and do not work within the cell. And we know that those frame shifts are expected to be pathogenic. That is really strong evidence that this is clinically actionable. We know this occurs in the middle of the gene. Genes are broken up into segments called exons. Within that region of the gene, a variant like this will likely be clinically actionable. We do not see this variant in large human or healthy human populations, indicating it's rare. This disease is rare, so we would not expect individuals that are healthy to actually have this genetic change at large numbers. And so we also can look at additional databases and resources online, and we can see that other clinical laboratories actually interpret this variant as clinically actionable. And then in this example case, the patient is known to have a personal or family history that's consistent with hereditary breast and ovarian cancer syndrome, which is associated with BRCA2. So within this example report, we also provide these personalized risk estimates, and these include your lifetime risk of hereditary cancer associated with that particular gene, which in this case is BRCA2. Um, Individuals will have, depending on their sex, will have a potential increased risk of female breast cancer, an increased risk of male breast cancer compared to general populations, which are represented in a light gray bar, and the gene specific cancer risk is presented as a dark gray bar, increased risk of ovarian cancer, and increased risk of pancreatic cancer. And if we look at these cancers that affect males in BRCA2, we can see this increased risk of breast cancer, prostate cancer, and pancreatic cancer. And then, based on professional society guidelines and also scientific evidence, there are particular surveillance strategies that are recommended for individuals that have clinically actionable or pathogenic variants within this gene. These include a self breast exams, clinical breast exam, mammography for male breast cancer, prostate specific antigen testing, or a digital rectal exam to look at prostate cancer or prostate cancer surveillance cancer. Of genetic tests, and in this case, risk card, is we look at individuals with cancer and individuals without cancer that may have a strong family history. For individuals with cancer, it can explain the primary driver of that individual's cancer. It can direct treatment options, inform surgical decisions, determine risks for other cancers to direct management, and provide really valuable information for at risk family members. For people without cancer, it can inform their lifetime risk of developing cancer. It can allow that early intervention and screening, which is very, very crucial. It can reduce unnecessary screenings for individuals that are negative, and can help guide family planning. So, one thing that's really important that we've touched on throughout this is family history and the effect that genetic testing can have for family members. So while you may have, as a male individual with breast cancer, a positive test result, it's not just you that's affected. Your siblings, your parents, your children have a 50% chance of actually inheriting that genetic change, either from you or transmitting it to you. And so, one thing that may be considered is familial cascade testing, and that's the process of testing at risk, close family relatives for that variant that you carry. And that can be really impactful for early cancer detection and prevention for your relatives. It also can enable detection of true negatives, which can be very psychologically reassuring and also prevent unnecessary screening. Um, and as I said, it depends on your relatives what their risk is. First degree relatives, like I said, your parents, your children, your siblings will be at a 50% risk. When we get out to second degree, grandparents, aunts, uncles, um, grandchildren, the risk drops down to 25%. And every degree you go out, it will drop by 50% more, or halve, I should say. 50%, 25%, 12.5%. Um, Testing is not recommended under the age of 18 because these cancer syndromes are typically adult onset. Um, but some of the genes on the panel are associated with increased risk of autosomal recessive conditions in young children, and that's why you want to consider family planning. Just to finish on this, because we talked about genetics and we talked about family risk, a big part of this is actually knowing your family history, especially for male breast cancer. And I gave a plug on this last year, and I want to do it again this year. Is, use a day this year or a day around the holidays, a day around Thanksgiving to actually talk to your family about their health history, because you may be surprised at what you find and you may actually be able to help each other. And so I just want to provide you with some resources, and I would highly encourage you to do this with your family members these holidays. And with that, I'll take any questions. And I just want to thank you so much for having me here. It's always a pleasure talking to you all.
We can't thank you enough, Hannah. Very, very, very good. Um, I have a quick question. Lately, we have been seeing so many younger men with breast cancer. Do you feel in the genetic scene? And again, I when I talk to them, when they search us out and they find us, first question I ask them, have you had genetic testing? Many of them say no, and I urge them to go get it done. I said, talk with your doctors. This is something you need because it's your family members, just like you said. They're the ones that need to know and to be checked and everything. One of the other speakers we had stated that lately they've seen that even at age 14 the youngers could be tested. What's your opinion on that?
Um, so I will say I have to be careful because I work for a company.
Yes, yes.
um, I I haven't seen data at the moment that's saying that there's an increase um, in the number of cases, and I haven't seen that with the prevalence data. But what I like to think is that organizations like yourself and social media posts and just through communication, people are starting to become more aware of genetic components to disease. And I'd like to think that it's starting to become more accessible. When we think about genetics, really, the human genome, the first draft was only mapped in 2000. And while we say that's 23 years ago, when you compare this to some other medical procedures, this is actually very, very new
Yeah,
technology.
yes.
And we're still, even now, learning just so much about the human genome. We sequenced it 23 years ago, and every day there's It feels like there's a new variant, there's a new technology or a technology enhancement coming out. And so to me, this is all still a very, very new field. And I think a lot of people still unaware about it. And we need organizations like you to make people aware, to make them start being health advocates for
Yes.
themselves. Check your breast, check for lumps, be aware of your body, notice skin changes. It's not just about, male breast cancer is important, but I think we all are getting that message. If we want to have a long, healthy life free for disease, we need to be stewards for ourselves and also our family members. So I'd like to think that social media and just increased education and awareness is driving this, and that's exactly what we want, Peggy, because the best time to get testing is when you're pre-symptomatic.
Yes.
From my perspective,
Totally. Yes.
because that gives you the option to start that. The earlier you start that surveillance and medical intervention, the better your outcomes will likely be. And then you have the ability to personalize your treatment and own that lifetime risk.
Totally agree. And Brett first found his lump when he was 17, taking doctors over 7 years to finally test him for breast cancer. Uh, and that was after he had over a year and a half worth of discharge out of his nipples that he mentioned to his father, but never told a woman. So guess what? The men didn't even know that that was a
And
sign.
do you know what? I think I'm going to drive home one point you just said then
Yeah.
is, and I'll go back on this slide, it's talking about it.
Yeah.
We need to talk.
Yes.
And we shouldn't feel ashamed about nipple discharge or lumps in our breasts. It should be a normalized conversation that we can have with people close to us.
Totally agree with you.
And I think that's also how we can change this story.
Yeah.
It and we can own it, and we can have some semblance of control. There's always going to be so much we can't control with cancer, but I also say this is why
Yes.
the health surgeon general has been making these big pushes every year. Talk about it, because by talking about it, you can help each other, you can help yourself.
Oh, totally. Several of the men we had talk about it, that when they, call, they talk about it, and then I'll get on a call with them later. I'm dealing with two brand new ones this week. And I said, one thing you need to know is that if you've got kids, if you've got grandkids, the first thing you need to do is say, we all need to be checked. We need to genetically be tested because, in Brett and our case, yes, we have no genetic mutation, okay? But this last go round, not only do I have Brett with breast cancer, I have over 16 first cousins, all with breast cancer, rest being women. But the bottom line is with your all's testing, because that's what we just did it again last year. It said there is something there that perhaps we still to be discovered, which is causing your family breast cancer. And I totally believe that. I know there's something out there in our families because, and I share this with you, my parents were second cousins. When you get that close, we're Lebanese background. And in Lebanon, you were fixed up with who you were going to marry the minute you hit the earth. And there are so many other nationalities out there, the Italians, everything. This was what was done back in the early years, okay? And we can't change that. That's still us. But as the genetic testing gets better and better, which it has over the years since when Brett was first diagnosed in 2010, oh my God, what it's doing to allow our families to be aware and understand their bodies. I tell every man, if you find something, do something. Don't just sit back and think it will go away, because guess what? It won't. And I've said this many of them. You're the one that's going to wind up going away if you do nothing. So, um, that's the hard facts of it. But yeah, what you all are doing is freaking amazing, and I can't thank you enough on behalf of my family because we talk about it. We don't not share with each other, and we learned that from Brett's situation. So
And I have to say, organizations like Male Breast Cancer Happen, you also advocate and push people to talk about it,
yes,
and you bring me in to talk about it. You bring
that's
in
right.
other people to talk about it, and to me, this is where the change, where we can drive change, and
Yes.
that makes me very, very happy, and I am just so privileged to be able to join you.
Yeah, well, we can't thank you enough. And these will live on in our YouTube channel. So anyone that ever has missed it, they're able to watch it, share it with family members, and that's the key. Share it with family members. Don't be afraid to talk about it because guess what? Men have breasts too. And
Yes,
my motto is together we will change the world because not one damn one of us can do this alone, guys.
exactly,
So
exactly.
I thank you enough for being part of our conference. And until next year, I say, but within this next year, we have our monthly meetings, and we'd love to have you back again when you have
I
something new you want to talk about.
or I would love to come and just listen. And as I've said to you many times before, knowing and hearing from patients that are behind those test reports that we sign out, it is just so crucial and so validating for the work that we do. And so I would love to reconnect with you.
That's awesome. We can't thank you enough. Appreciate it.
All right.
Okay.
Have a